Welcome to G Fun Facts Online. Let's start with a fact that might surprise you. If your ancestors came from anywhere outside of sub-Saharan Africa, then you are part Neanderthal. That's not a metaphor. It's a biological reality written into your DNA. For decades, we pictured Neanderthals as our brutish, primitive cousins, who were simply outcompeted and replaced by smarter, more advanced modern humans. An evolutionary dead end. But this picture was completely wrong. A revolutionary science called archaeogenetics has torn up that old storybook, revealing a much more intimate and complex history. By learning to read the faint genetic whispers left in ancient bones, we've discovered that the ghosts of these extinct relatives are not gone. They live on inside of us. This is the story of how we found the human-Neanderthal hybrids that walk among us today. For a long time, reading the genetic code of an ancient human was pure science fiction. DNA, the blueprint of life, is incredibly fragile. The moment an organism dies, its DNA starts to break apart, degraded by time, water, and microbes. After tens of thousands of years, you're left with a puzzle made of billions of microscopic, shattered pieces, all mixed in with the DNA of bacteria and fungi. It seemed like an impossible task, but science finds a way. The revolution truly began in the 1980s with the invention of a technique called the polymerase chain reaction, or PCR. You've probably heard of PCR from modern medical testing, but its power is in its ability to take a single tiny fragment of DNA and make millions or even billions of copies of it. Suddenly, the microscopic amounts of genetic material surviving in ancient remains were enough to study. A Swedish biologist named Svante Pääbo was a key pioneer. In 1985, he managed to sequence DNA from a 2,400-year-old Egyptian mummy, proving it could be done. But the real target was our much older relatives. To start, scientists focused on something called mitochondrial DNA, or mtDNA. Think of it this way. Most of your DNA, your nuclear genome, is packed into the nucleus of your cells. It's your main instruction book. But your cells also have tiny power plants called mitochondria, and these have their own small, separate circle of DNA. Critically, while there's only one nucleus, there are thousands of mitochondria in each cell. This means there are thousands of copies of mtDNA, drastically increasing the chance that some of it will survive the ravages of time. In 1997, Pääbo and his team hit a major milestone. They sequenced mtDNA from the very first Neanderthal specimen ever found, the one from Germany's Neander Valley. The result was electrifying. The Neanderthal mtDNA was clearly distinct from any modern humans. This suggested our lineages had split a very long time ago. At first, this seemed to support the out of Africa with replacement model, the idea that modern humans swept out of Africa and completely replaced archaic groups like Neanderthals with no intermingling. But mitochondrial DNA is only a tiny part of the story. It's inherited only from your mother, so it can't tell you anything about your father's side or the rest of your vast genetic family tree. To get the full picture, scientists needed the Holy Grail, the entire Neanderthal nuclear genome. In 2006, Svante Pääbo, now at the Max Planck Institute in Germany, launched the Neanderthal Genome Project. This was an incredibly ambitious undertaking. His team had to invent new methods to piece together a full genome from degraded, contaminated fragments of bone. Their main samples came from the Vindija cave in Croatia, where Neanderthal bones around 38,000 years old were exceptionally well preserved. After 4 years of painstaking work, in 2010, they published a draft of the Neanderthal genome. The results, published in the journal Science, sent shockwaves through the scientific world. They compared the ancient Neanderthal genome to the genomes of modern humans from different continents. A stunning pattern emerged. People whose ancestry was from outside of Africa Europeans, Asians, Native Americans, and Pacific Islanders, all carried between 1 and 4% Neanderthal DNA. People with purely indigenous African ancestry, however, had virtually none. The conclusion was undeniable. Modern humans and Neanderthals had interbred. The old replacement model was dead. The story was now one of leaky replacement. As our Homo sapiens ancestors migrated out of Africa around 60,000 years ago, they encountered Neanderthal populations, most likely in the Middle East. And they didn't just fight or ignore each other, they had children together. These early human-Neanderthal hybrids then continued their journey, spreading across the rest of the planet and carrying that Neanderthal genetic legacy with them. Just as the world was absorbing this revelation, the same technology led to a discovery that was, if anything, even more shocking. The story moves to Denisova Cave, a remote shelter in the Altai Mountains of Siberia. This cave is a remarkable place, a crossroads of ancient history, occupied by different human groups for hundreds of thousands of years. In 2008, archaeologists found a tiny fragment of a pinky finger bone. It was so small that it couldn't be identified visually. So it was sent to Svante Pääbo's lab for genetic analysis. The result was jaw-dropping. The DNA from that tiny bone didn't match Neanderthals, and it didn't match modern humans. It belonged to an entirely new, previously unknown group of archaic humans. They were named the Denisovans, after the cave of their discovery. When the full Denisovan genome was sequenced, it added another layer of complexity. Not only had Denisovans and Neanderthals interbred with each other, but Denisovans had also interbred with modern humans. Today, traces of Denisovan DNA are found primarily in populations in Melanesia, people from places like Papua New Guinea and surrounding islands, who carry as much as 5 to 6% Denisovan DNA. This suggests that as modern humans spread east across Asia, they met and mixed with Denisovan populations. Denisova Cave was a prehistoric metropolis of sorts, a place where at least 3 different human groups, Neanderthals, Denisovans, and modern humans, coexisted and interbred over millennia, leaving a tangled genetic web we are still trying to unravel. For a while, all this evidence was statistical, based on comparing ancient genomes to modern ones. But then, scientists started finding the ancient individuals themselves, the direct proof of this intermingling. In 2012, again from Denisova Cave, came the analysis of a bone fragment known as Denisova 11. The team nicknamed her Denny. She was a girl, at least 13 years old, who died around 90,000 years ago. When her genome was sequenced, the scientists were stunned. Denny was a first-generation hybrid. Her mother was a Neanderthal, and her father was a Denisovan. This was the first time anyone had ever found the direct offspring of 2 different archaic human groups. It was the ultimate smoking gun. And Denny's story had another twist. Her Denisovan father also had at least one Neanderthal ancestor further back in his own family tree. This meant that these encounters were not just freak, one-off events. Then there's Oase 1. In 2002, a 40,000-year-old modern human jawbone was found in a cave in Romania. Morphologically, it looked like a mix of modern and archaic features. In 2015, genetic analysis confirmed this suspicion in the most spectacular way. The Oase 1 individual had between 6 and 9% Neanderthal DNA, far more than any modern human. The segments of this Neanderthal DNA were also very long, which is a key clue. DNA gets broken up and reshuffled with each generation. Long, unbroken chunks mean the interbreeding event was very recent. Scientists calculated that Oase 1 had a full-blooded Neanderthal ancestor just 4 to 6 generations back in his family tree. This was incredible. It proved that interbreeding wasn't just a single event that happened in the Middle East 60,000 years ago. It also happened in Europe much more recently. Interestingly, the Oase 1 lineage doesn't seem to have survived into modern populations. suggesting he was part of an early pioneer group of humans in Europe that eventually died out. But even before DNA analysis, there were hints. In 1998, in Portugal's Lepido Valley, archaeologists found the nearly complete skeleton of a 4-year-old child, ritually buried 28,000 years ago. The Lepido child had a modern human chin, but a stocky, robust body and lower leg proportions that were distinctly Neanderthal-like. It sparked a huge debate. At the time, without genetic proof, many dismissed it. But in light of everything we know now, the Lepido child is seen by many as a powerful symbol of our shared past. So what does this ancient inheritance actually mean for us today? What traits did we get from our Neanderthal cousins? It's a mixed bag of advantages and disadvantages. Remember, when modern humans left Africa, they were entering new continents with new climates, new diets, and, crucially, new diseases. Neanderthals had been living in Eurasia for hundreds of thousands of years and were already well adapted. Interbreeding with them was like a genetic shortcut, a way to quickly pick up useful adaptations. Some of the most important genes we inherited are related to our immune system. We got variants of genes called toll-like receptors, which are a critical part of our body's first line of defense against bacteria and viruses. These likely gave our ancestors a huge survival boost. But there's a downside. That more aggressive immune response is also linked to a higher risk of allergies in modern people. That's right, your hay fever might be partly your Neanderthal ancestors' fault. Our skin and hair were also affected. Neanderthals passed on gene variants that influence keratin, the protein that makes up skin and hair. These likely helped our ancestors adapt to different levels of sunlight in colder temperatures outside of Africa. Neanderthal DNA has also been linked to a whole host of other traits and health conditions. For example, a major genetic risk factor for severe COVID-19 was inherited from a chunk of Neanderthal DNA. Other variants have been associated with an increased risk for type 2 diabetes, Crohn's disease, and lupus. One variant that increases blood clotting may have been great for ancient hunters who needed wounds to heal quickly, but in our modern sedentary lifestyles, it increases the risk of stroke. Even our brains may be subtly influenced. Certain Neanderthal gene snippets have been linked to a higher risk for depression and even nicotine addiction. Now, it's crucial to understand that these are just statistical links. Having a particular Neanderthal gene doesn't mean you will definitely get a disease. Your health is a complex dance between thousands of genes, your environment, and your lifestyle choices. But it's undeniable that these ancient encounters left a subtle but significant mark on our biology. The science of archaeogenetics has completely redrawn our family tree, but it has also opened up a universe of new questions. We know interbreeding happened, but we don't know how. What were these encounters like? Were they peaceful exchanges of people and ideas, or were they violent? How were hybrid children treated in their communities? The archaeological record offers tantalizing clues. In some parts of Europe, it seems modern humans and Neanderthals lived in the same regions for up to 2,900 years, perhaps long enough to share technology and culture. And the careful burial of the Lepido child, stained with red ochre and laid to rest with pierced shells, hints at a level of care and symbolic thought that we once thought was unique to our own species. The future of this field is even more exciting. As the technology gets better, we'll be able to analyze even older and more damaged DNA, perhaps uncovering more lost relatives. The story of human-Neanderthal hybrids is a powerful lesson. Our past is not a simple ladder of progress, but a messy, tangled, and beautiful web of connections. The Neanderthals did not simply vanish into extinction. They were not a failure. A part of them was absorbed into the great river of human DNA, and it flows within billions of us today. The ghosts in our genes have a story to tell, and for the first time, we are finally able to listen.